Okay, welcome back to part 2, where we're going to discuss a little bit on RNA, how ribosomes work, and the history of DNA discovery, a particular study of 50s misogyny. Now, this, of course, is incredibly relevant for a later video that I'm going to do, which is that we've now moved into an era of having RNA viruses. So they've been in the news, RNA vaccines, rather, and they've become really, really popular in the news, particularly with the announcement of the Pfizer vaccination in the last week, and of course the other one today, which works on similar technology. Okay, so we'll come to those in another one, but it's really important to understand how those work, that you understand basically what RNA is. So before we get too far into it, I need to explain to you that a little bit, and we're not going to go into this in massive details, about how DNA is actually read and understood. You know from previous knowledge that DNA holds the information for making everything about you, right? You also know that ribosomes make protein. RNA is one of the crucial molecules in making A talk to B, okay? Making the DNA talk to the proteins, making the DNA's instructions become protein, okay? So it acts as a messenger and a translation molecule and is actually integral in the ribosome itself. So this is our system of transcription and translation. The DNA never leaves the nucleus, just like all good blueprints should never leave the office. So copies have to come out and be made into protein. They have to be read and made into protein when the cell needs that particular protein. And they also have to be used as adapter molecules and so on and so forth. So that system there we'll come back to in much greater detail later in the course. So what is the structure of RNA? Well, it's still a nucleotide, the nucleic acid. It's still exactly the same as DNA in terms of it's still got phosphates, it's still got a base there, although they're slightly different. Remember, they're U, not T, uracil, not thymine, but it's a ribose sugar. So on that carbon 2, where the DNA just had hydrogen, there is an OH group here, so it's ribose sugar, okay? And still got a sugar phosphate backbone. That construction of the backbone, the sugar phosphate, 5' to 3', that's still the same. Okay, still got bases sticking out, but normally it would be single-stranded. So let's move on then. Ribosomes themselves, okay, and this is something that students regularly forget, which you should remember, are made from RNA and protein. They are made of protein for stability and ribosomal RNA because RNA can bind to stuff. Those bases can actually bind onto things, okay? So that's the two components. It's just something you have to know, that ribosomes are made from RNA and protein. Okay, this was a question which students generally got wrong. Very easy. Give the two types of molecule which ribosome is made. Okay, and there's the mark scheme. One of RNA, ribonucleic acid, nucleic acids, anything like that, and protein, polypeptide, amino acids, anything like that. Okay, so both for one mark. You just have to know it. Okay, so in comparison, DNA versus RNA. This is also a very common question. Okay, so we've talked about the bases, that uracil instead of thymine, that methylated group I was talking about at the end of the last video. You can just see there when it isn't here on the uracil. That's the only difference structurally between thymine and uracil. Cytosine, adenine, guanine, same thing here. So RNA, single-stranded, DNA, that famous double helix, usually. Viruses are always weird. Okay, so viruses sometimes have double-stranded RNA, but more on that at a later time and in a different video series. So if you are asked, and I recommend you write this down, make a note of it, go away and learn this if you want to get an A-level in biology. If you're just watching for entertainment, then fine, carry on, you strange people. DNA versus RNA, the key points are like this. DNA is usually much longer. Now, a reason I've highlighted and underlined and bold there is you can't say long. DNA is long, RNA is short. What do you mean? There is a comparison between the two. Long in terms of what? Long in terms of your fingernails? Long in terms of a suspension bridge? What are you talking about? It is longer than your average strand of RNA, which is shorter. So put the ers on. These are comparative statements. Okay? DNA is usually double-stranded and RNA is usually single-stranded, but they're usually there because viruses are often an exception. DNA, as we've said, the deoxyribose, that deoxy comes from no OH group at carbon 2, whereas in RNA you do have an OH group at carbon 2, which is what makes the RNA much less stable. OH groups are reactive, right? They form condensation reactions with things. So DNA not having that is much more stable. It's It's also why any vaccine made of RNA has to be stored at minus 80 degrees C, as has been put out in the news quite a lot in the last week or so. So we've said DNA contains thymine, RNA contains uracil, and then the uses, what are they for? Well, DNA is an information storage molecule. It's a long-term, large, stable information storage molecule, a heritable molecule that passes from generation to generation. RNA has three different functions. One is for carrying messages between the DNA, between the nucleus and the ribosome. We call that messenger RNA or mRNA. One is for translating the information on mRNA into the language of amino acids. So that's called transfer RNA, tRNA. Okay, more on that later. And as we've just said, some of it is for forming ribosomes. That's rRNA or ribosomal RNA. And if you can remember all those, that'll pretty much set you up for comparisons between DNA and RNA. Okay, so let's have a little bit look at the history. Okay, now this was in the 1950s, so we had different teams competing to discover the structure of DNA. Okay, now the fact that there was something in the nucleus that was important was known from very, very long time ago. In the Crimean War, there was a surgeon who was looking at the pus in soldiers' bandages and noticed all this strange material within nucleuses of the cell, and he called it chromatin. Okay. We then worked out fairly quickly that it was a combination of this sugary stuff, this ribose sugary stuff, and protein. For a long time it was thought that the protein must be the information carrying molecule because it's more complicated, but then there were various groups who came around to the idea that the DNA was the most important molecule, and then raced to try and structure it. Now Watson and Crick were working out of Cambridge, and Rosalind Franklin and Maurice Wilkins working in London. Now Watson and Crick were brilliant, absolutely. The other ones credited with the discovery of DNA, but they were also known to be a little bit lazy in terms of doing the actual experimental work. Whereas the group with Franklin and Wilkins in it was there working away doing a process called X-ray crystallography. And that's what this famous picture here is. Now this was one of Rosalind Franklin's pictures that she generated. Now the way X-ray crystallography works is you take the molecule that you want and you suspend it in a crystal of something salty maybe, right, and you put that on a little spike. In the 50s it was literally inside a paint tin with photographic film around the inside, and you fire X-rays at it. The molecule scatters the X-rays and they form a pattern on the photographic film. Okay, from that pattern you are then supposed to work backwards with nothing but a chalkboard and your brain, right, from the pattern back to the structure of the molecule. Now this picture here was incredibly important. The clearest picture, that X shape, okay, was incredibly important. Rosalind Franklin took this picture, but it stayed with her for a long time. She didn't necessarily know exactly what it meant, but she knew it was important. Now Wilkins here, there was a very much of an animosity between these two for various reasons, and you can go, there are many documentaries and many books written about this whole thing. He for right or wrong, and it's not standard scientific process, passed this picture taken by Rosalind Franklin to these two, and it was the last piece in the puzzle. Okay, they'd already claimed once that they'd figured out the structure, called these guys up to Cambridge to see it. Franklin laughed at them, went, I don't know what's right, but I know you've got that inside out. So this was the last piece of the puzzle, passed, pre-published, unpublished, from Wilkins to Watson and Crick, who managed to put together the final structure of DNA. I'm not taking anything away from Watson and Crick. They obviously they were the people who made the mental leap to put all the information together to get that initial structure, but it was Franklin who had done the hard graft. And the things about X-ray crystallography is it uses X-rays, and in the 50s there wasn't a lot of shielding from those X-rays, and it was ultimately those X-rays that at a very early age killed Rosalind Franklin through cancer, which meant that these three received the Nobel Prize for physiology, and Rosalind Franklin got nothing. Okay, she has since been highly honoured, and next to Marie Curie, she is probably the most famous female scientist out there, which is a damning indictment of our noting of female scientists in our society. But it still, goes down as one of the most sort of potentially controversial stories of a major scientific discovery in history. Okay, so if you want to look into it, It's a fascinating story, very interpersonal story about the very human aspects of science. You can have a look at some of these documentaries: DNA's Opening Pandora's Box, Life Story, The Race for the Double Helix, and DNA Secret of Life. You can have a look at those. They are all great. They're a bit old, okay, but that's good because it means that these guys, apart from Paul Franklin, these guys are actually in them, okay, so you can hear from the horse's mouth what their impression of what went on was. Okay, so I do recommend you go and have a look and decide whether you think that Watson and Crick were rightly awarded and went down in history as the discoverers of DNA, or whether Franklin was the victim of massive 1950s misogyny, or if it's a little bit of both. Okay, so that's a bit of the history. Okay, from this, right, we've now come to this. This is where we are now. These pics. This is the first direct image taken a couple of years back of DNA, which since the discovery we've done amazing things with DNA, and it wasn't until we could get nanoparticles to support a single strand of DNA that actually we can see, right, just about that 10 nanometer rotation of the double helix. Okay, so we've basically come full circle from this image being fought over through to all the incredible genetic discoveries of the last 50-60 years, through to this image here, which confirms fundamentally that the inferences from this were absolutely spot on. Okay, so that's where we've come to. Right, how is this going to pop up on an exam paper? So explain how the structure of DNA is related to its function. That one there would be a long answer question, 4 to 6 marks. Okay, and the key to answering that question is explain. So you're supposed to use your own knowledge. How? Right, so literally linking the two things together. Don't just say things about DNA and expect that to get you marks. Say things about how that DNA links to its job. Okay, so explain how the structure of DNA relates to its functions, so its job. Okay, if you want to have a go at that, pause the video, give yourself, I don't know, 5-10 minutes, have a go at that, and then come back and we'll look at the answers. Okay, so I'm assuming that you've done that. Here we go. We've got 6 possible marks here. Sugar phosphate backbone for 1, or double-stranded, provides strength and stability, protects the bases, and protects the hydrogen bonds. It's a very long molecule, so it can store lots of information, as we said. It's much, much longer than most RNA. The helix makes it compact. Right, the base sequence allows information to be stored, and sequences which code for amino acids and proteins. It's double-stranded, as we mentioned at the end of the last video, so its replication can occur semi-conservatively. Now, you probably haven't done semi-conservative replication yet, so look at another video on that. I'll explain what that is. Basically, the strands can both act as templates, so you can get 2 cells from 1. You've got weak hydrogen bonds for replication, unzipping, strand separation, many hydrogen bonds in there, so it's very, very stable or strong. Okay, so if you want to pause and have a look at your answer compared to that, see what you would have got. But please note how each of those answers is linked to a function. So they said a thing about DNA, and then this word "so" in bold. Okay, so there's a reason for all the structures. Right. Last few ones here we'll have a look at. So the following figure represents part of a DNA molecule. Draw a box around a single nucleotide. Well, you should be able to do that if you have a phosphate, a sugar, and a base. Then you're doing well. Okay, Figure 1 shows one base. Figure 1 shows one base pair of a DNA molecule. Name part F of each nucleotide. Well, you see those pentagons there, and we should all know they are correct. That is the deoxyribose sugar. Now the key there, look, if you attempt to put just sugar or ribose, it says Figure 1 shows one base pair of a DNA molecule, not an RNA molecule, a DNA molecule. So you know that that sugar must be deoxyribose. Okay, and they want that specificity at A level. Now this is a classic question which some people get straight away, some people need pointing out, but once you know what they're looking for, it's really, really easy. So scientists determined that a range of DNA that sample of DNA contained 18% adenine. What were the percentages of thymine and guanine in the sample of DNA? Now you might think at first looking at that, there's not enough information there, but there absolutely is. Okay, so if you know that there's 18% of adenine, you know there's got to be the same amount of thymine, because A and T bind together, right? So 18 plus 18, 36. Okay, What does that leave? That leaves 64% left over, and you know that must be split equally between the cytosine and the guanine. Okay, so therefore you can work out the percentage of thymine as 18, right, and then you can work out the percentage of guanine as 32. Okay, so now you know how that's done. It pops up quite regularly. Right, that's the end of part 2 of our structure of nucleic acids and nucleic acid topics. I'll try and do more of these on DNA replication and transcription translation as time goes on, but I hope that was helpful. See you soon. Bye bye.